All right, we ready? So I updated the slides on the biotechnology tissue culture to include a few things. I just wanted to show a couple of those. But we were talking about this last time about the idea that Agrobacterium tumefaciens can be used as a tool essentially to introduce your gene of interest. And so this is a new slide. So here, normally, if it's just living its normal life, it can cause what's called crown gall disease, which is what you're seeing here. And you've probably seen that in your life already, just out walking around, hiking. It's found everywhere I've lived, I've seen it in the environment. And so basically it's causing this overgrowth, this local overgrowth that looks like a tumor because it has a plasmid that has genes that code for the auxins and cytokinin type hormones. And that allows for this cell division, cell growth, and you get this growth, but you don't get any kind of differentiation at all. And so they call it a gall or a tumor. And basically what the scientists figured out is if you can remove those genes so that you don't get this localized growth and you don't have that kind of worry as the plant is growing up. If you can get those out of the plasmid and then put your gene of interest into the plasmid and then put that plasmid back into an agrobacterium and just make it grow up in the lab, you know, get lots of it so you can get a lot of the plasmid. And then you start mixing the bacteria in with some plant cells in the lab. And as they're mixing, you know, usually you do it in a broth culture and you give a little bit of a shaking so they're bumping into each other. And then the agrobacterium will infect the cells and put the plasmid in to the plant chromosome that has your gene of interest, but it won't be causing any issues like this. So you can end up with a really healthy plant. And this is what it normally does. But when you use it in that way where you knock out those genes and put your gene in, you don't, and then you grow the plant up through tissue culture and you'll have a fully healthy plant. You won't need to worry about it. So, but you can see it, this crown gall disease happens on herbaceous plants too, and like rose bushes. If you ever seen rose bushes, you can see that. You can see these are quite petite, but then you can see some that can get quite large.
So is it infected with the bat? So do we infect the bacteria and then the bacteria infects the tree and that's how we get the gene expression?
Yeah, I actually work in tissue culture. And so, because usually when they say, I'm gonna put this gene into this plant, they want every cell of the plant have that gene. And so you have to start with just a handful of plant cells in a tissue culture, which is intubation in the lab. And so basically you get the bacteria to sort of plasmid it into the plant cell. And then you coax those plant cells to grow into a full mature plant. And then every cell in that plant will have your gene. Maybe with drought resistance, disease resistance. And but you definitely need to use tissue culture to, like, you couldn't just go to a tree and inject, you know what I mean? You have to start with cells and then grow it up into a mature plant.
I guess.
Yes, it would stay local. It wouldn't be too beneficial for the plant.
Violence that infects us in that changes that's why I was curious if they would do it the same kind of.
Yeah, it's, if you want, generally scientists want every cell to be infected and then they know they have to resort to tissue culture.
Yeah.
So. Let's see. So I introduced or added those slides in. And then, oh, the other one I added in because here we went through this yesterday and listing out, and I forgot to put in a slide on what I meant by the liposome. So let me just cover that as well. And so the liposomes, you might have heard that term already because it's the idea of, they use this in human medicine. And so this is the idea where you create some kind of phospholipid bilayer vesicle. And then you can easily do that in the lab. And then, so that's what you're seeing here, the liposome. And so it's just a vesicle, a phospholipid bilayer vesicle. And then you can put your drug of interest in it. And then what happens is the phospholipid bilayer of the liposome will easily be able to get through the cell membrane and deliver the drug. And so they're doing that in human medicine, but they're also doing it in the plant world too. And basically though, when you work with plants, you need to, again, you're resorting to tissue culture ideas. And typically what you do is remove the cell wall again of the plant cells. So you're working with the protoplast idea of the plant cell so that it's easier for the liposome to fuse and get brought in to the cell. And so this is a paper that was published last year. And just to give you an example of why they might be doing this in the plant world. And so here they're looking at introducing selenium. And so they often can call them nanoliposomes just because they're so tiny. But basically, they're putting selenium, that element, into the liposome. And then in this case, what they did was they sprayed it on a foliar spray onto the wheat plants. And selenium is a nutrient that humans need and plants need. And so the idea here was to see if this would be effective in boosting the plant. And it did. I mean, I'm not going to go into details on the paper, but if you just look at selenium concentration, they were able to get quite a bit. And they were tracking to see, you know, is it showing up mainly in the leaves or shoots, or is it getting transferred down to the roots? But anyways, this is the control. And you can see a huge difference. So very successful trial that they did. So that's just one example of why they might want to work with liposomes in the plant world. But it's really, I think they're more further along in like human medicine with this idea of working with liposomes. So it's probably a thing you'll hear about more and more.
Or is it just for helping the plant grow?
Well, there's all kinds of ideas that they're coming up with, like how can we use this to improve plant growth and get higher yields? So yeah, I mean, with this idea, with the selenium, it's kind of a 2-for-1 because it helps the plant, but then when you eat it, it helps you too. And so it's, you know, something, so there's, well, America has some problems with this too, but certainly other parts of the world, there's like key nutrients that are missing in people's diets that it can be very serious, cause very serious illnesses and even death. And so this has been an issue of how do we beef up, so to speak, some of the plants that they'll be eating to have more of that. And so this is one idea that they're looking into. So I think it's a great idea and it's a great delivery system. So in this case, they worked with a full plant. You know, they didn't have to do the tissue culture stuff. They just sprayed it onto a mature, you know, a growing plant in the field. So that's nice too. It's pretty straightforward, and hopefully people that don't have a lot of technology in third world countries, you know, they could do this and it would help. So really promising for those types of ideas. So I wanted to put that in there. So that's in there now too. Okay, and then we were looking at, I think we left off, oh, flavor savers. That's where we left off. So okay, so now this next thing is new. So here's a cute little mouse. So believe it or not, they use mice sometimes in the plant world. And so they gave it the cute name Plantibodies. So we have antibodies, so do mice. Antibodies are part of our immune system, right? And they kick into gear when we get sick with something and make antibodies against whatever it is that's attacking us. Maybe it's a bacteria infection. Maybe I was just talking about strep throat in my other class. That's caused by bacteria. Has anyone in here had strep throat?
Yeah.
Yeah, isn't it awful? Oh my goodness. It's like it, yeah, you swallow and it's like razor blades are stuck in there just slicing at you and ugh. But anyways, if you have strep throat, your body is producing antibodies to fight that or virus and so forth. And so what they do, they capitalized on this idea where they use mice and they will expose the mouse to a plant pathogen and wait for the antibodies to get produced. And then they figure out what gene in the chromosome of the mouse is making that antibody. And then they take that and put that into the plant, usually using agrobacterium as a tool to do that. AB is antibody. And so they'll get the genes that make that particular antibody that the mouse, so this is all done in the mouse. They'll figure out what gene it is. And then they'll transfer that gene to the plant. And then when the plant gets exposed to that same pathogen that they exposed the mouse to, the gene turns on, makes the antibodies, and you can get the pathogen. It won't cause disease. Yes.
Is that like the GMOs for like
Yeah,
corn
it,
and stuff?
it would be a GMO because they took a mouse gene. So definitely genetically modified. And that in itself is a whole nother topic, like the pros and cons and the issues with that. And I haven't kept up fully with the field of all the genetic modification that they're doing. I'm sure it's pretty extensive with plants. But I think, in my opinion anyways, I take every case on a case by case basis. So I don't do a blanket statement saying GMOs are all bad or GMOs are all good. It's like, well, let's look at it. What plant? What did you do? Where was it inserted? There's a lot to it. So yeah.
So, could like artificial selection in terms of like how we change wild mustard to be like kale?
Broccoli.
Oh my God, could that be considered genetic modification because we're changing gene expression?
Here, I know what you mean, but they put that under the umbrella of selective breeding. Because the humans selected for these traits and did it through sexual reproduction. And so they could only do it through compatible species, you know what I mean? Like they couldn't do something wild, like it wouldn't fall under this. This would be genetic modification because we're taking a gene from a mouse and putting it into a plant rather than saying, I'm going to take this cultivar of the corn plant and this cultivar and put it together and see what the offspring looks like. And so like that's what they did with the mustard. They kept playing.
How strict your method.
Yeah. So the this right. Yes. And so, one thing about genetic modification is you can take, so I think let me go back to, where is it? Where did it go? Oh, here. So where it says breeding methods using sexually incompatible germplasm. So that's basically the idea of genetic modification. You're taking genes from two different species that could never reproduce and give viable offspring. They're too far apart. They're different species. And in this case, with the mouse, I mean, you're two different kingdoms of life. So the idea of selective breeding, they're still compatible and you can get them to breed and create offspring. They'll just have interesting traits like the broccoli and cauliflower. Okay. So liposomes and then, plantibodies. So it is kind of a wild idea, but it works. And they've had some good success with that. Whoops. Did I put in the So they said, ag crops, I'm looking in the notes section of this slide. Ag crops such as tobacco, tomato, potato, soya, bean, alfalfa, rice, and wheat are commonly used for production of plantibodies. And so they have been trying it out. So I have more information in the notes section if you want a little bit more about that. So when you look at what's going on in the biotechnology realm, a lot of what they're focused on is, okay, we have, what's the population of people on the planet right now? Where are we at? 8 billion? And don't they say it's going to be 10 billion by 2050 or something like that? So, so that's a lot. And, it's kind of scary if you go to a plant biology conference and start talking to people who grow food. And they're like, wow, what are we going to do with 10 billion people and how are we going to feed them? You walk out of those symposia a little alarmed. And so this is one avenue, there's many, but this is one idea is, okay, we have to figure out a better way to help the plant survive to harvest, hopefully grow really vigorously, produce a lot of edible portion of the plant. And so some of the biotechnology ideas that we've been talking about, this is what they're doing. They're looking at helping the plant fight pathogens, get better yield. They're looking at how do we increase the nutrition of the edible parts per gram of tissue, just make it more nutritionally dense, which I think is a great idea. And so we are pretty much out of usable land. Like we're already there. We're capped out of good land. We have marginal land that we can buy, grow crops, but again, we're going to need to tweak the plants to deal with the marginal land unless we figure out a way to remediate the land a little bit better. So for example, the rice plant, which is, feeds billions of people every day. And they're trying to figure out, I think I told you, they're trying to make it a C4 plant. And so again, this would be biotechnology that they would be using. They're looking at human medicine too. They actually are thinking about ways of using biotechnology to create drugs for, cancer drugs and things like that. Edible vaccines, again, that's the idea of, especially third world countries. If you go to vaccinate people in third world countries, you're often way out in a rural area. You're not near any technology or refrigerator. Like so many of our vaccines, you have to keep them cold until they're injected into the person. And so that's, believe it or not, that's a huge stumbling block. Like, how do we vaccinate? Yeah, they're using potato.
Yeah.
Yeah, they're doing potato. Yeah, they're using the potato plant. Yeah.
Stable.
Yeah, because they can grow the potato and eat it. And yeah, and so they're looking at that. So it's, yeah. So yeah, it's really,
dystopian, but cool.
Yeah. So, and again, third world countries are lacking in key minerals. And so, looking at ways to beef that up. So increasing iron levels in the rice, and they're trying to increase the protein levels in rice. Vitamin A is a huge problem in third world countries. A lot of countries, people don't get the vitamin A. And so you might have heard of golden rice. I'll show you a picture, but that was developed quite a bit ago, actually, to provide a good dose of vitamin A. And again, if you have salty soil, which some of our marginal land is pretty salty, and so they're looking at, okay, can we get plants to grow in it, even though it's not the best land? And then, of course, could we use plants and plants are pretty good at remediating contaminated soil. So is there a way that we can tweak plants to be better at it, even better?
Question, was there any way we can make like a natural water source into a hydroponic system? Does that make sense? Or we're able to grow plants within
Actually, in India, they are doing hydroponic growth in rivers and lakes.
like a food?
Yeah, like they have like rafts.
Yes.
Yeah. And then they'll put, like wells in it, and they put the plants in it and grow the crop. Right in like the rivers, slow moving rivers normally. Yeah. Yeah, so they're looking at that. In fact, there's a lot of really good research going on in India with, growing plants in rivers and.
It's all in certain areas.
And then also in
All
polluted areas too, require remediation. They use the aquatic plants just, kind of soak up the pollution. Yeah, so there's a lot of good ideas there. Here's a picture of golden rice. So this has been beefed up with vitamin A. And vitamin A, you don't want deficiencies in it. I think in the yeah, so golden rice was designed to produce vitamin A, the precursor beta carotene. Okay, so this idea of taking genes from two different sources where they cannot, produce any sexual, they can't go through sexual reproduction. So that's the fancy incompatible germplasm phrase. So that means you're working with different species. And so they often phrase it across species boundaries. So if they start talking about that, what they're thinking about is we need to use biotechnology in order to accomplish whatever we're thinking. And very often, one strategy that they've tried too is this idea where they'll strip the cell wall off of two different species. And they're like, what would happen if we then try to get them to fuse? And then their chromosomes are mixing together, like what would happen? And so they've done that in tissue culture. So again, you work in a liquid, a flask with liquid in it with all the correct nutrients to support their life, but you get protoplasts from two different species and mix them together. And they actually do merge. You usually have to encourage it with either a chemical like polyethylene glycol or that electroporation idea, but then they fuse. And so then it's like, okay, what do you do? And then if you can get the technique down, you can encourage that fused, those two cells that fused to go ahead and grow into a plant. So hybrid fusion is the technical term for that. So hybrid fusion means you've taken protoplasts from two different species. And if you can get it to work, then they start talking about somatic hybrids, which, that's the plant. So you start saying, oh, that's a somatic hybrid plant. All right, so if you can get it to work. So it's a plant that will carry genes from two species that no longer can like breed with each other. It doesn't work. But through this process, you get it to work. So probably the most classic example of that is this, where on the left is Indian mustard plant, and on the right, I think that's cress, and in the middle is the somatic hybrid. And this experiment, they were looking at the soil is contaminated with too much heavy metals. When we have a lot of soil like that, unfortunately, where it's heavy metal contamination. And so this represents a wide cross between the plants. This crossing was done by protoplast fusion. Indian mustard on the left tolerates lead, but not zinc or nickel, whereas stonecress on the right tolerates zinc and nickel, but not lead. So like, well, what would happen if we put them together? And so this pot has all three, all three pots have all three heavy metals. And so you can see both of these are struggling because they're only partially able, resistant. Whereas this one got all the traits. And it's like, yeah, I can handle lead and zinc and nickel. And so this was one of the first success stories with this idea of a somatic hybrid. It's pretty bizarre. Yes.
Are the hybrids able to reproduce?
Generally, no. I don't think they produce viable fruit. I don't know that they've got that to happen. I have not heard of it myself, that they can go that far. But for ideas like this, where you just want to remediate the soil, if this plant is taking up the heavy metals and you don't really plan to eat it, you're just using it to decontaminate. So in that regard, it's successful. So somatic hybrids. And then somatic embryos is another idea where you can, if you got the technique down in tissue culture, you can take, so somatic means just regular body cells. Like most of your body is somatic cells. And then you have your gametes, which form egg and sperm, right? So a plant has that too. They have a bunch of body cells, okay? That normally don't do any kind of, like with egg and sperm, eventually you get an embryo. Here, a somatic embryo, it's like, that's what? Somatic should never lead to an embryo because they're just normal body cells. They're not sperm and egg. But in tissue culture, if you know how to do it, you can take some just normal plant cells, like leaf cells, and get them growing in tissue culture and encourage them to cluster together like embryos do. When the sperm and egg fuse and then you got cell division, you get a little tiny cluster of cells that made you, finally you emerged, but that's the idea. And so this, when they this to work, that cluster of cells behaves like an embryo, a plant embryo. And then what they did was they took the somatic embryos and created artificial seeds off of that. So here you have the somatic embryo. Once they get that, then they cover that to protect it with this gelatinous matrix. And then they refer to that then as an artificial seed or synthetic seed. Synseed is another cute little jargon word for that. And so they're starting to do this a little bit more, but basically you can buy artificial seeds from certain companies, and they will typically say on their website, it's a somatic embryo that they've created. One story that was published, when was this? A couple of years ago, cannabis synthetic seeds, an alternative approach for commercial scale of clonal propagation and germplasm conservation. So they're coming up with these interesting ideas. I have never run this technique, so I don't know exactly how they encourage those cells to become and behave like an embryo. But they're getting it to work. And it seems really far-fetched. I bet the first time they tried it, they were like, yeah, this isn't going to work. But then plants are amazing and they got it to work. So they will look at expanding this. I know I've seen a few tidbits out in the news saying that they're looking at this more seriously. So anyways, something rather bizarre that's going on in the biotechnology world. Okay. So that's all I have time for with the biotechnology stuff. Any last questions on that topic right now, anyways? Okay. So the next idea is we're shifting gears now into looking at all the different phyla of plants on the planet. But before we do that, we're actually going to start with fungi. Yay, which is one of my favorite topics. I even wore, can you tell what my earrings are? Wore my mushroom earrings today. So in both my classes, I'm going to start talking about fungi today. It's like, oh, yay. This is like best day ever. So we're going to get into fungi. So traditionally in a botany class, intro botany class, they would cover fungi. And that was way back when they thought fungi were plants. Nowadays, we know that's not true, but we still cover it because, well, first of all, I love fungi. And second of all, they're so important for plants, both good and bad. There's good and bad fungi that interact with plants. I'm gonna focus on the good ones because it would take forever to talk about all the bad ones. There's, of all the microbes that can cause problems for plants, fungi win, unfortunately. However, the flip side can be said too. There are beneficial fungi that are have been vital for plants since the minute they crawled out of the sea, so to speak. And so we'll talk a little bit about that too. This is a little snippet of a poem about mycorrhizal fungi, so we're going to talk about that as well. And so in lab tomorrow, right? Tomorrow's Thursday, you'll be setting up your edible mushroom kits. And then you'll be asked to watch a video on endophytes. And so that's what I'm going to talk about right now. First, we'll get into endophytes. And so endophytes, there's plenty of fungi that are endophytes. Endo means within. P-H-Y-T-E means plant or something plant-like. So it's fungi that live inside of plants, but aren't causing harm to the plant. And so when you hear the word endophyte, it has a positive spin on it. And there's also plenty of bacteria that are endophytes too. All right, so it's not exclusive to fungi. There's all kinds of bacteria that hang out inside of plants. But for this section, we're going to focus on the fungi. And so they don't, so really the definition, at least the way I define an endophyte, is that they live pretty much their whole life inside the plant. And they don't cause disease to the plant. So mycorrhizal fungi, when we get into that section, I don't qualify those as endophytes because so much of the fungus is outside the plant. It's inside too, but a lot of it's outside. So I'm doing a more strict definition where it's inside the plant for pretty much its whole life. So this is, did I show you this picture ever? I can't remember. This is a leaf tissue on tissue culture media. And so all of this growing out of the leaf tissue is fungal endophytes. So in that tissue culture medium, there's some sugar in there and minerals, and the fungus is like, well, I like that. And so it's starting to grow out of the leaf onto the tissue culture. So this is what I talked about with tissue culture, how you can't really control, you can't kill off the endophytes because then you'd kill off the plant as well. And you run this risk unless you put in some kind of antifungal into the tissue culture media. But it's a nice image to show how the endophyte is there and it's growing out, in this case, nicely visible. So they live in the plant tissue. And from a fungus's perspective, it's kind of nice because they have a home, a nice stable home. They're protected from getting eaten and attacked by other things. They get access to nutrients. The plant is, available for nutrients, but not to the point of being pathogenic and harming the plant. And then the more they look at what's going on with endophytes, the more they discover new ideas, like what is the endophyte doing for the plant? And some of the research is definitely the endophytes help the plant defend themselves. It can be directly or indirectly against pathogens. And so sometimes the endophyte produces a chemical that's very strongly inhibitory and can kill the pathogen. And so the plant will, in return, keep that fungus alive and shelter it, and the fungus can fight off the pathogen. And so that's a nice win for the plant. They've seen nice studies where herbivores, bunny rabbits, deer, mammals of any kind come along and eat the plant, that the fungal pathogen can discourage that practice, and by producing certain chemicals. They've looked at, can the presence of the endophytes help a plant deal with drought or too hot conditions, too much salt, toxins? And the answer is yes, 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 and yes. So again, they're finding that this partnership between the endophyte and the plant is, and there's many endophytes. It's not like just one fungus is in there. There's all kinds of fungi that can be in one plant as well as all kinds of bacteria. Yes.
I don't know if I'd be like mycelium, or is that completely different?
Yeah, so the fungus is, so when we talk about fungi, like all of this that you see, you would say, oh, look at the mycelium of the fungus. Isn't that pretty? And so that's the one way in which fungi grow. They have this long kind of hair-like or filamentous growth form, and that's what we call the mycelium. And it's also called hyphae, and we'll talk more about that. And so that's how they grow in the plant. They're growing in between the, like in the cell wall areas, they're growing like that in the cells. Or in this tissue, sorry, in the tissues. So it is also the idea that the endophytes, especially in the root tissue, can help the plant get better access to minerals, better uptake of minerals, so that the plant is getting nicely fed that way. And so, like I said, the more they look, the more they're finding that there's all those advantages to having these endophytes. And so the video that you'll see, and you'll do that on your own time, not in the lab, but the video is Dr. Gary Strobel, who used to be a plant pathologist out in Montana, I think Montana State University. And he kind of pioneered this research of, he didn't really discover endophytes. He wasn't the first to discover them, but he was the first to say, well, let's see what they're doing. Like, how are they helping the plant? And can we capitalize on that in any way? And so it's a very interesting video of his saga. He was controversial because he cared so much about plants that he did something a little illegal. But I could totally relate to why he did it. And unfortunately, the grant that was funding that was through like the USDA or something like that. And they're like, no. And so he got in a lot of trouble. Anyways, so it's quite the soap opera. So you can watch that and form, I'd be interested in what you think about it. If you think that, they were right and he was wrong, or vice versa. But anyways, that very stressful period of his life, he said it was incredibly emotionally and mentally draining, but that it really got him into this path of, let's look more carefully at endophytes. So he made lemonade out of lemons, and he recovered and ended up with a very successful career. Unfortunately, he's recently retired. I've had several botany students go, I'd like to do a master's with him or something. And he just recently retired, but I think his son is working on the East Coast he's a professor out there, and I think he's still doing some endophyte work. And I'm sure there's others doing endophyte work now too. It's gaining. So the question is, if they're in the tissue of the plant, how did they get there? That's one big question. And so with the endophytes here, they realized that a lot of it is passed down, so to speak, through, vertical transmission from one generation to the next in the seeds. And so here you're looking at, seed germinates and grows into the plant. And what happens is that seed can have endophytes in it. So what happens is the endophytes are kind of packaged with the plant embryo. When that gets planted and the plant grows, then the endophyte can actually move. So it doesn't, just stay in the seed and that's it. Once the plant starts to grow, the endophyte can migrate into different parts of the plant body. And then when that plant goes through reproduction and is about to make fruit and seeds, again, the endophyte moves into the seed. And so you can get the endophyte pretty much persisting with that plant through generations. And so this is something that I think were kind of surprised to see, that that's a pretty regular occurrence, that there's endophytes inside the seed. There's also, of course, all kinds of microbes that hang out on the seed. I think I might have mentioned that at one point, that now more people are being a little bit more thoughtful, because usually you surface sterilize seeds if you're a grower. Because if you have 10,000 seeds you need to plant, you don't want them all, like, what if they're all infected? You're gonna lose your whole crop, which is understandable. So they would surface sterilize their seeds, but now more people are going, yeah, maybe we shouldn't do that, because maybe these microbes are supposed to be there. They're part of helping that seed and that embryo survive and grow and thrive. And so there's all kinds of ideas about surface microbes too on the seeds and what we should do. So I just wanted to stress that, once an endophyte gets into a plant, so through seeds is one way to get this to happen. A lot of other endophytes, they think, primarily are coming from the roots, which means the soil. So when you think about, the soil is chock full of microbes, especially near the roots. And so some of those microbes just stay in the soil, some are on the root surface, and then there are some that can go into the root, maybe through a tiny wound, something like that. And they can gain entrance. And if the plant allows it, then it can become an endophyte. And so here you're seeing some images that they did where they're showing how the endophyte got into the roots and then started migrating to different parts of the plant. So once it gets into the root, that's not necessarily its final destination. And so that's an important concept because sometimes you do want those endophytes in like near the flower. Because maybe that will help the plant defend itself against attack in that area. Or maybe it'll help with nutrient supply to those areas. And so if you're a grower, you want the plant to produce flowers and then eventually fruit. And so being able to kind of protect the plant perhaps by a beneficial endophyte.
The endophytes or can they go into like the water and then do like the water absorption? Can they go through with them as well? Or is it?
Yes, yep, they can go that way for sure. So up through the xylem, yep. They can go through a wound of some sort. You can get in that way. What about leaves? What would be a way to get in? Yeah, the stomata. So good bacteria and fungi, bad bacteria and fungi can get in that way. So some of Duncan's research we're very curious about the endophytes that he might have had in his hemp plants that he grew. And so we're trying to hunt for those currently. And we're looking at leaf tissue and flower bud tissue. And because he's very interested in use of biocontrol agents and do they have any impact on CBD production, CBD levels. And so we're looking at endophytes as part of that too. But anyway, so it's important that you understand that it's not like, oh, they're only in the roots. No, they can get into the roots pretty easily because there's so many microbes that are there. Chances of getting in is pretty good, at least for some, not all, but some. And then they can start migrating through the plant body. So this is some work from Gary Strobel's lab. And you can see these are all beautiful fungi that are growing on agar plates. And you can see that there's, everything that looks different is different. They're different species. And he was given the nickname by some of his friends as the Indiana Jones of fungus hunters. So he went all over the world. When I found out they had made a DVD about his experiences, I emailed him because it wasn't available anywhere. I can't remember how I heard about it, but anyways, I emailed him and I said, hey, I'd be very interested in your DVD. Could I buy a copy? Because I teach and I'd like to have the students learn about this. And he's like, oh yeah, I'll pop one in the mail. I'm on my way to the Amazonian rainforest. I'm like, okay. So he did. He popped one in the mail. I got it a few days later. But he said, yeah, I'm catching a plane, So he's traveled all over the world and he gets grants. Wouldn't that be a nice job? He gets grants and then that funds him to travel around the world looking for endophytes. So that was a huge part of the last stage of his career. And so you'll see in the video how he travels to different regions and he's hunting for endophytes. So, and then they take them back to the lab. They try to isolate them, purify them, grow them up, and then start studying them and looking at what kinds of interesting chemicals might they produce, what are they doing? And so he's been able to, he was part of the team that figured out this drug, the paclitaxel. And it's the world's first billion-dollar anti-cancer drug. That's what it looks like. They've never been able to figure out how to synthesize that in a lab. So they discovered this in yew trees, Taxus species, yew trees. Well, plant biologists started to get really worried because they were chopping down yew trees left and right to get this drug. It's $1 billion drug. They're gonna keep chopping down trees to get this. And so Gary Strobel and another colleague said, well, I wonder if the yew trees might have an endophyte that's actually producing this and not the yew tree, which would be great, because then if you could isolate the endophyte that makes this, much easier to grow that up in a lab, and you can make that drug in huge concentrations. It's not that hard. Then you can stop chopping down the yew trees. And so thank goodness they thought to look, because that's what happened. There's a fungus that makes this, it's called Taxol and they sell it, but paclitaxel. And so now they just use the fungus. They don't have to keep chopping down the yew trees. So that was hugely successful. And I think that's what really kind of lit the fire of, hey, you know, endophytes might have a ton of interesting aspects to them. Yeah, this is an incredibly powerful and successful drug. It's for certain types of breast cancer, and it can put you into total remission. I mean, it's that successful. So it's, you know, you can understand why they were chopping down the yew trees left and right, because it was incredibly successful. But thankfully, so what they discovered is that, did I put that in here? So the host plant and the endophyte can produce the same product. So what the yew tree is producing this, but the fungus is too, okay? So the host plant and the endophyte can produce the same product, but what's really exciting is that you can also find endophytes that produce novel drugs or chemicals that the plant isn't doing. And so then it's really interesting to start thinking about, well, what are they making that for? What is that doing for the plant? And then, of course, Gary Strobel's, a lot of his work is, if we can find out what these drugs are, maybe they have human medicine applications or, you know, medicine for other animals. And so, and then they discovered that one endophyte can actually produce more than one drug, more than one interesting chemical. So this has been a big push for novel drug discovery and looking for like anti-cancer, antiviral. They're looking at, you know, stuff like blood pressure medicine. And I mean, the list goes on and on and on and on. And somewhere in the notes I have, in the notes section, I have some, yeah, this slide, I have a bunch of, you know, antioxidants, insecticidal, anti-diabetic. And so they're looking at endophytes for this. And so, the idea that, so plant physiologists are a little, I think, when they figured out endophytes exist, they were a little dismayed, because they always just said, oh, this plant is making this chemical, you know, and then they realized, oh, it's fungi and bacteria doing it too. And so it's kind of muddied the water on who's making what chemical and why are they doing it? Because you know, it's like, well, maybe the plant's doing it, but actually maybe it's their endophytes that are doing it. So it's a little confusing. But anyways, great idea for drug discovery and just helping the plant exist. We got to, you know, feed all these billions of people. And one quote that I read in one of Dr. Strobel's papers, he was saying, stop cutting down the tropical rainforest, because when you do that, it's not just the tree or the plant, you're losing all those endophytes as well. So good quote. All right, we'll stop there.